Hey everyone, we're going to move on to the cell cycle, which includes both mitosis and cancer. Throughout the chapter, you'll be seeing this diagram up here in the upper right-hand corner. That shows the cell cycle, and you'll see mitosis, which we do often talk about, is actually just a very small part of the cell cycle. Keep that in mind. Here's something for you to ponder. Biology is the only subject in which multiplication is the same as division. So when a cell multiplies, doesn't it have to divide? Interesting thought. All right, let's start off with a little story. The story of Henrietta Lacks. This is Henrietta Lacks right here with her husband. And this is Henrietta's son. She did have a few children, and this is one of her sons. What does this have to do with mitosis? Well, if you've ever heard of something called HeLa cells, H-E-L-A, these are cells that are still around today that actually belong to Henrietta Lacks, even though she died quite a long time ago already. She was a victim of cervical cancer, and before she died, and when she was being diagnosed and treated for her cervical cancer, the doctors took some samples, and those samples grew in petri dishes in the lab and actually turned out to be like some cells that grew indefinitely and grew very well outside of the human body in that petri dish. And so those cells are still around today, even though she has been long gone. So there's her son showing her picture. And we have a lot to thank for Henrietta's donation. They've used those cells for a ton of research around the United States, maybe even around the world. One example is helping to find the polio vaccine. So why are her cells so resilient, and why do they grow so well outside of the body? Well, number one, they're cancer cells and they're growing uncontrollably. They're not listening to like any cell signals to tell them to die and go through apoptosis. They're just continually growing, and they're able to repair their own DNA so that they don't really age. And remember, they are cancerous cells, so they're a little bit different and behaving different than normal cells. But what they do have in common with normal cells is that they are going through a cell cycle and they are going through mitosis. It's just that they're doing it so much more quickly than regular cells do. And we'll often refer to Henrietta throughout this chapter and remember her and her contributions. So once again, there's that chart, the cell cycle. When we take a look at the cell cycle, there's this whole thing around the outer edge called interphase. And interphase is divided into G1, S, and G2. Then there's a small little window or piece of the pie where mitosis happens and cytokinesis, which that's called the M or mitotic phase. Right? When a cell divides, or should I say multiplies, it makes two daughter cells. And we expect that the daughter cells, when it goes through mitosis, are genetically the same as the original. So where did it all begin? Well, right there, as an egg, right? The egg is like one of the biggest cells in the human body, and you can almost just see it with the naked eye. So it started after fertilization and then began to grow by mitosis after that, and went from 1 cell to 100 trillion cells in just 9 months through dividing or multiplying. Why do cells divide? Well, reproduction is a definite to make more cells, especially if you're a single-celled organism. Think of it that way, that's the only way that you have to reproduce. In us, as a multi-celled organism, when our cells divide, we call that mitosis, but when it's in like an amoeba or a bacteria or prokaryote, we call it binary fission. Cells divide for growth, like in organisms like us, and for repair and renewal, such as that poor little lizard over there who's lost its tail and will now do some mitosis there to grow a new one or to repair what happened with that injury. When we go through the cell cycle and things like cell division, make new cells, we have to consider what are some of the major players from the cell in those processes, right? So definitely the nucleus, where you have your chromosomes and DNA, because you're going to have to make copies of that stuff so that the daughter cells have the chromosomes and DNA, because that's the instructions for the cell to make all the proteins that that cell will need. So daughter cells must have those instructions. So the nucleus, as like the protector of the DNA and chromosomes, is a major player in the making of new cells. Then the cytoskeleton, which includes all those little protein fibers and filaments and microfilaments and microtubules that make up the spindle fibers and such. All of that is part of cell division in the cell cycle. When I think of the cytoskeleton and all those little proteins, I like to think of a building with scaffolding like this, because they make up that infrastructure that gives the cell its shape and definition. But then also, when it's ready to divide, it helps with that as well. So the nucleus protects our DNA. The structure includes a nuclear envelope, like a lipid bilayer, but it's a double membrane there, and it has some pores, because there are things that will travel through the nucleus, such as mRNA is one thing that has to go through the nucleus. Ribosomes are made inside the nucleolus and have to travel out of the nucleus. So things have to get through. Just some examples. There's an example of a DNA wrapped around these little proteins called histones that make up our chromosomes. Those are all housed there in the nucleus. So there it is, and you can picture it with the ER right outside of it with the ribosomes. I like that picture. Move me over here. All right, and then taking a look at the cytoskeleton, the structural support of the cell, all these little protein fibers from microfilaments, intermediate filaments, and microtubules. When you look at it with an electron microscope, look at that. So when you think of a cell and you think there's cytoplasm, that it's all juicy inside, well, yeah, but there's also these filaments inside and microtubules that give everything shape and support, and also allow a cell to be able to move. They're responsible for the motility and connect to any outside structures that are responsible for motility, like cilia and flagella. Cilia are the little hair-like structures. The flagella is more like the long tail like a sperm would have. They also organize some of the activities of the cell, including mitosis, when a cell gets ready to split in two. So it looks a little bit like that. Connecting, giving everything shape and support. Now there's these structures that we really haven't paid much attention to until now. Now they are going to be key components of the cell. These are called centrioles. They look like little rod-shaped stars. Basically, they are bundles of microtubules, and you have a pair of them in each cell after division. That pair will actually double, so you'll have two pair, and they will move to opposite sides, and they're going to help to pull that cell apart. So very important in the cell cycle and in cell division. So now just some vocabulary here. The whole area of where the centrioles are found is called a centrosome. So this whole yellow area with little proteins poking out, that's a centrosome. Again, there's one of them after a cell is divided, but it does double and makes two. And then the centrioles are within the centrosome. Right? So these are going to help set up the spindle fibers that are going to help pull apart the cell. End of our little cell tour and talking about the players involved there. So we got to get the right stuff. So what is passed on to the daughter cells? Well, you have to pass on the exact copy of the genetic material, or as close as possible. I know it's not always perfect. There's mutations in there and such, but you want it as close as possible. So DNA does double or replicate shortly before cell division, before mitosis. So you also want to pass on to your daughter cells plenty of organelles like mitochondria and ER and things like that, plenty of cytoplasm, membrane material like the phospholipids, enzymes and proteins that cell is going to need to survive and function normally. And those things are divided during cytokinesis. So mitosis is the actual division of the nucleus that divides the DNA or chromosomes in half, and cytokinesis is the division of the rest of the cell, the division of the cytoplasm that will divvy up the organelles and membrane material and molecules and enzymes and such. So cyto means cell, kinesis means movement. So the movement of the rest of the cell that divides that up. So the M phase in the cell cycle will consist of both mitosis, the splitting of the nucleus, and cytokinesis, the splitting of the rest of the cytoplasm. So let's do a little overview here. I'm going to have to move myself out of the way. So we have some drawings of some cells. So I want you to remember IPMAT throughout this. So I stands for interphase. This is what a cell looks like in interphase. That's kind of the longest part of a cell cycle, where a cell is normally doing its regular stuff, growing and making molecules and doing reactions and things like that. But when we're going to get ready for mitosis and cell division and cytokinesis, then the cell will begin to enter prophase, which looks like this. There is kind of an in-between phase. I don't often recognize it, but some textbooks do. It's called prometaphase. It's an in-between phase, getting you to metaphase, which is probably one of the most recognizable phases of mitosis, because all those chromosomes are lined up in the middle at the equator, and then anaphase when they're being pulled apart, and then finally telophase and cytokinesis when our nucleus is reforming and cytokinesis pulling the rest of the cytoplasm apart. So that's an overview. Let's take a look at each of those a little more specifically, starting with interphase. And here's that diagram. I love this. When we say interphase, just think of a cell saying, I'm working here. I'm doing my regular stuff. I'm making enzymes. I'm doing cell reactions. I'm growing. I'm making organelles and things like that. So that's 90% of the life cycle. So that goes all the way around this edge here to show that interphase is most of what the cell cycle is. All right, and let me go back. Interphase consists of these little parts, and actually jump to this one. G1 is the first part. You can say it's called gap 1 or growth 1 phase. Again, the cell is doing its everyday stuff and growing. The S phase, S stands for synthesis. This is when the cell has received a signal to make more DNA or replicate its DNA. So this is when the chromosomes are copied. Again, think back to chapter 11 here. Think of how cell signals arrive, dock with their receptor, or maybe the receptor is in the cytoplasm. It depends, but think chapter 11 here, that to do these things, the cell must receive signals. And then there's the G2 phase, or gap 2 or growth 2 phase. This is where the cell is getting ready for mitosis and cell division. And so we're putting the finishing touches on everything, the final growing, adding organelles and cytoplasm and such, proteins, enzymes, and all these things that a cell will need to be successful. taking a look at the cell cycle here, a normal cell goes through this, like epithelial cells. Those are skin cells, blood cells, stem cells that are in your bone marrow, which are making like red blood cells and white blood cells. So they go through this life cycle where they will end up going through mitosis and making more daughter cells. But we've got some other individual cells in your body who don't quite go through the cycle like the rest of the normal cells do, which is kind of interesting. So what cells are those? Well, brain and nerve cells and muscle cells. It seems after development, after you've become a fetus and you're born as an infant, you don't grow more heart cells or more brain cells or nerve cells. You've kind of got what you've got. So they go off into what's called this G0 phase. They don't divide ever again. For instance, muscle cells. You don't make more muscle cells when you build muscle. You just add more microtubules and microfilaments, more of those protein structures to the cells you already have and make them bulge out more. And that's why this is also why when you get nerve and brain injury or heart injury, it's hard to come back from that because you don't make more cells to replace any that might be lost. Liver cells are kind of interesting. They're in the middle. They can go back and forth. They can enter G0, but then they can come back and be like a normal cell and go through regular cell division like the rest of the cells do. So this is why you can actually donate a piece of your liver to someone and yours will repair and grow, and so will theirs. Interesting stuff, the way these cells do these things. All right, so with interphase, if I look at a cell under a microscope in interphase, I can actually tell that, oh, that cell's in interphase by what it looks like. When I look at the nucleus of that cell, maybe if I was to stain it, I wouldn't see any chromosomes in that nucleus. The chromosomes would not be formed yet. That happens more towards mitosis. So when the DNA is not formed into chromosomes, it looks more like this. Kind of looks like spaghetti and meatballs, really. And this is called chromatin during this time. Here I can see, though, that there's two sets of centrioles or centrosomes that contain centrioles. And so I know mitosis is on the horizon. It's coming up. Right, so when a cell gets a signal that it's going to be time to divide or multiply, coming soon, then it'll go through S phase, which is the copying and replicating of the DNA. How it does that, remember A's matching with T's and C's with G's, we'll be getting to that in some future chapters very soon. But we want to make sure when we copy the DNA that we get as close to possible as we can exact copies. The error rate seems to be about 1 per 100 million bases. So in a 3 billion base pair mammalian genome, you would have about 30 errors or mutations to your somatic or body cells in there. So I guess no copy is perfect, right? Remember, when DNA is copied, it's called semi-conservative, that the strands separate, and so you have your parent or old strands, and then you fill in new daughter strands. So part of the DNA that you're giving to your daughter cells, part of it has the parent strand, part has the new strand, so you call that semi-conservative. All right, so just a little reminder on how DNA is organized. DNA is organized into chromosomes, because having a long 3 billion base pair strand of DNA, that would get all tangled up and be all confusing. So DNA gets wrapped up very tightly around these histone proteins to get it all nice and organized, and then makes 46 chromosomes in a regular somatic body cell. and then looks like this, makes an X shape. So I can tell that a chromosome is duplicated when it makes an X shaped. All right, so there it is, copying and winding. So here's a mitotic chromosome right there. So this is all going to happen during that S phase. So looking more closely at a chromosome, the dark purple, that's one chromosome. But its copy or its sister chromatid is the light purple. So they're connected here at what's called the centromere. There's these little protein fibers sticking out. That's what the mitotic spindle will grab onto to be able to pull those chromosomes apart. Those are called the kinetochores. So when I count the number of chromosomes in an individual, I count the number of centromeres. So even though this is 2 chromosomes here, or what we call sister chromatids connected together, I'm still just going to count that as 1. I know that does get a little confusing. So this is what they really look like under an electron microscope. A trained eye would be able to take a look at this and tell that, oh, this is chromosome number 1 right there, and there's chromosome 2, or there's the X, and there's the Y. Maybe someday you'll have a trained eye. So this is called a mitotic chromosome. So remember, you have 46 chromosomes in your somatic or regular body cells. Half of those came from your mom, half came from your dad. So let's say the purple ones came from mom and the green ones came from your dad. So 23 from mom, 23 from dad. Normal chromosomes look like this picture down here in the bottom left corner. But during the S phase of mitosis, they double and form what's called sister chromatids, or the X shape, or also sometimes called tetrads as well. So again, here's the purple one that came from mom and the green one that came from dad. We'll say this is chromosome pair number 1. And they have the same genes on them. Maybe they both have the gene for blood type or something like that. And so they call these homologous chromosomes, the one that came from mom that matches up with the one that came from dad, that are in your own cells. And you'll pass these on to your offspring someday. Which one will you pass on? You can only pass one of them on, remember. You can't pass them both. So your cells will go through meiosis to get that all divided up, but that's another chapter down the line. But again, these are homologous chromosomes. They contain sister chromatids in this picture. And then take a look up here. This is the picture, electron microscope picture of a mitotic chromosome right there. It looks awesome. All the winding of that DNA around those histone proteins to wrap it all up nice and tight. And this is, remember, going to be pulled apart during mitosis. So it starts with this, then it looks like that, and then ends like this in the daughter cells. All right, so these are called karyotypes. And a karyotype is basically just a picture of your chromosomes. This is a female. You can see all 46 of them here. And remember, half of them came from your mom, half came from your dad. Scientists are getting better at knowing which one is which. And this can tell us a lot about the offspring, or tell us a lot about you, whether there's going to be any abnormalities and such. So these chromosomes are removed, and then they're stained, and then matched up in pairs, and then that makes the karyotype. Then down here we've got the X chromosomes. They are lined up from large to smallest. We've got the X chromosome though, but that's our sex chromosome. So there's thousands of genes on there, and some have nothing to do with sex, but some of them do, and so that's why they call it the X chromosomes. If you're a female, you have 2 of those, and if you're a male, you have X and a Y. So with a female, you have all homologous pairs, but as a male, the X and the Y are not homologous. They do not have the same genes on them. the X has its genes, and the Y really only has maybe less than 30 genes on it. It's a pretty small chromosome. So we'll be talking about what are the implications of only having one X and one Y further down the line. All right, I do want to say again, these are matched up and lined up from biggest to smallest, except they kind of made a mistake at one time. And actually 21 is the smallest pair of chromosomes, but they never went back to fix it. So it stays this way. If you have 3 copies of the 21, that's called Down syndrome. So that's one example where you can live with more chromosomes than just having 2 pair. When you have 2 of each chromosome, you're called a diploid individual. D-I-P-L-O-I-D, diploid individual. And many mammals are diploid. All right. So let's go to the M phase here, that little piece of the pie with mitosis. And mitosis is divided up into 4 main stages: prophase, metaphase, anaphase, and telophase. Mitosis actually means the dance of the chromosomes, believe it or not. And you can remember this by saying PMAT. All of these phases are recognizable under a microscope. So during prophase, if I was looking at this cell under a microscope, your chromosomes would start to become visible. I would see them, that they're doubled, that they're in the form of sister chromatids, or the X shape. I would see that the centrioles are doubled, and they're starting to move apart from one another, and the mitotic spindle, the little protein microtubules, are starting to extend from those centrioles. This is actin and myosin, the microtubules that form that, and they're able to contract. They actually make up your muscle cells as well. And so that's where we're going to be able to get movement to be able to pull this cell apart. Notice the nucleus is starting to go away, and the nucleolus as well is starting to disappear or go away. There's a little transitional phase here called prometaphase, where the nucleus has gone away more. Our centrioles have really gotten to opposite sides, and our chromosomes are very defined and starting to move. But metaphase, probably the most recognizable of all the phases, all those chromosomes are lined up at the equator, or the center of that cell. And attached on each side is the mitotic spindle to those kinetochores at the centromeres, getting ready to pull those sister chromatids apart. So each daughter cell will receive only one sister chromatid. There's what it might look like under an electron microscope there. Anaphase, also pretty recognizable as well, because our sister chromatids are being pulled apart, almost like they've been lassoed in their center and are being pulled, and their arms and legs are flailing backwards as they're being pulled to the opposite sides of the cell. How are they being pulled? Well, the biggest idea right now is that the little filaments are walking along the mitotic spindle. When I think of, I should go back, metaphase, think middle, anaphase, think apart, to help you remember that. So again, the movement, they're trying to figure out how this actually works. So if you think of like your fingers like this, that those protein filaments can move forward and pull upward and walk literally along that microtubule. And then the actual mitotic spindle then disappears as that walks along that, or dismantles. Telophase, pretty recognizable as well, because now our cell kind of looks like a figure 8 as the nucleus has divided. And in fact, the nucleus might be coming back as well. Our chromosomes are starting to go back to being chromatin again. So the They're unwinding from their chromosome. The nucleolus is reforming, if you're able to see that. Notice each cell now has only one centriole pair again. And we can see this indent, or what's called cleavage, that's happening to cause cytokinesis, or the actual division of the cytoplasm, to begin. So cytokinesis there, that's the division of the cytoplasm. So you get what's called a cleavage furrow. So think of it like this, like you're tightening a drawstring to pull those apart. This is a constriction belt of actin microfilaments. Actin has the ability to contract. So there it looks, like right here, like a drawstring tightening around the middle. So that's called the cleavage furrow. So you're going to end up with two daughter cells after this. Here's what it looks like in real cells. So if we look at fish cells, since they're in a clear shell, we can actually watch it happening. See it happening there. Notice how metaphase is so recognizable when those chromosomes are right in the middle there. I will say this is not the best anaphase. That even I would probably think this is still metaphase here, but they've got this as anaphase. I've seen better ones. And we've got the cleavage furrow happening here in telophase. This might be like late anaphase, early telophase on that picture. In plants, it's a little bit different because they have a cell wall to contend with. So basically what happens is a cell plate forms here, made out of membrane material that comes from the Golgi apparatus, or the Golgi bodies. And then that material will form the new cell membrane, and then a cell wall will be laid down in between there. So it's really not as dramatic as when you watch an animal cell divide and you see that cytokinesis drawstring tightening. With plant cells, it's a little less dramatic. All right, looks like that. These little bubble or vesicles coming from the Golgi, laying down membrane material to separate those two daughter cells. Here's some good pictures. This is a good anaphase right there. We even have a prometaphase picture. So notice we've got interphase, where you really can't see much with chromosomes yet, but you're starting to see them here with the prophase, prometaphase, and metaphase. They're all lined up in the middle, and in anaphase pulling apart, and then a nice telophase where even the nucleus is reforming, and you're just hitting the end of the cleavage furrow and cytokinesis. And this is an onion root. So an onion root is a growing part of the onion sticking down into the ground. And if you take cells from there, you can find many different cells who are in different phases of their cell cycle. Some of them, actually most of them, are going to be in just plain old interphase, because that's 90% of a cell cycle. But you're going to be lucky to catch some of them doing anaphase, some of them doing telophase, prophase, and such. So where did we get all these properties from? Well, it probably evolved from the first prokaryotes, who divide by binary fission. Prokaryotes have a circular chromosome with maybe just a few million base pairs of A, T, C's, and G's. So again, it's going to be even a shorter process in them than it is in a mammal. So they do double their circular chromosome. Remember, there's no nucleus to contend with here. So they do double that, and then they elongate the cell, make a ring of proteins, and then pinch in two. Remember, they also don't have membrane-bound organelles like mitochondria and ER, Golgi apparatus, things like that. So this happens a little more quickly than in, let's say, a mammal cell. So these are some of the leftover things from evolution. Oops, this toe will undergo mitosis. Get it? Mitosis for repair. Ouch. Donut mitosis. Nice. If anybody wants to make these and bring them in, I would love that. All right, so I've got lots of pictures here for the rest of your slides, so you can get a lot of practice at looking at cells in these different phases. All right, there's some more, and then I have the learning targets here. This set of notes did not cover all of our learning targets, mainly 1 through 6. All right, 7, 8, and 9 will be covered in another set of notes where we will look at regulation of the cell cycle and cancer. Biology, the only science where multiplication and division are the same thing. Let me know if you have any questions.